Hey guys, here is the part B of section 16.2. I think now we're actually going to do a part A, B, C, and D of part 16.2, section 16.2. Part A of 16.2 was on the semi-conservative replication and how they figured out how DNA replicates and that it does it with a general unzipping of the DNA, or take the DNA, two strands, make them separate, and then add complementary DNA strands to that. And so we're going to take a look at the specifics. This time it's going to be kind of an overview is what I have written here, and it's not a complete overview, it's just kind of some concepts that need to be understood before you can get into the super detailed part that we're going to learn about DNA replication in a little bit. But first thing we have to look at is, how does the DNA separate? If the DNA is going to separate and become two template strands that we can add new nucleotides to, how does it separate? And so it ends up that along DNA there's a bunch of spots, and what they're missing here, this is showing as it's starting to unzip. But if we were to look at DNA, so here's double-stranded DNA, and I'll put the rungs of the ladder here, just pretend these go along. Double-stranded DNA, this is horrible, there we go. There would be specific locations along here where each of these rungs of the ladder is a sequence, right? A's, T's, and C's, and G's. Well, there are specific spots along here where there are sequences, so maybe from here, let's change this, sorry, having trouble. Maybe from here to here, there's a sequence of A's, T's, and C's, and G's inside of this, and that sequence, basically, we call that the sequence an origin sequence, and it tells a series of proteins that, hey, this is the place where we want to start to unzip DNA. So oftentimes students get the idea that DNA, when it unzips, it starts at one end, would start at this end and just unzip the whole direction. Okay, that's not true. It actually unzips at a bunch of locations along the way. So you can see here, this original, the dark blue is the original strand of DNA, and there would have been an origin here, an origin here, an origin here, and those proteins, when they read those sequences, the origins, they say, okay, we're going to unzip. As it unzips, it creates these little bubbles, replication bubbles. And at this replication bubble, it's actually a series of proteins on this side that are unzipping in that direction to the right, and another series of proteins on this side unzipping to the left. So it's actually unzipping in both directions. These bubbles, you can actually see them if you're looking at DNA as it's being replicated in a micrograph, an electron micrograph, where you can see this is a DNA double strand, and here it splits into two strands and comes back together, and it's a single strand, and then it splits, and there's another bubble, and here's another bubble, and you can actually see these. That's how they were discovered. Those are the bubbles. Another terminology, and we're going to use it a lot, or another term, is a replication fork. If we look at the bubble and look at where this box is, that is a replication fork. Where it is unzipping, there's another replication fork here as well, where it's unzipping in that direction. The light blue that you're seeing is the new DNA being placed in here to match the old DNA, and you can see that we don't unzip the entire DNA molecule first and then add new DNA. What we do is, as we're unzipping it, we add new DNA. Okay, and so as this replication fork moves this direction, we're adding DNA behind it. You can see that progression here, and these two bubbles are working towards each other, this fork going this way, this fork going this way, and eventually they'll meet and we'll have a whole long length of replicated DNA, and eventually the entire DNA molecule is going to be replicated. So that's yeah, just the unzipping. Okay, that's the unzipping. Now we're gonna zoom in towards these replication forks and see what happens at these replication forks where we add new DNA nucleotides. Okay, here we're looking at if this is the parent strand of DNA right here, parent strand of DNA, and we have, this is unzipped, so it's no longer connected to the other template or parent strand of DNA. So here we have complementary nucleotides being added. So where there was a T on the template strand, we had an A nucleotide coming in. Where there's a G, we have a C, and so on. What we want to point out here is that when these nucleotides come in, and again, a nucleotide consists of three parts. You have a nitrogen base, you have a sugar, it's deoxyribose, and then you have a phosphate. Well, what we wanted to point out, what the book wants to point out, is one of the driving forces here has to do with that this nucleotide, it's not just one phosphate that is attached to it, there's actually three when it's by itself. They call it a nucleoside triphosphate. It has three, and if you remember from ATP, three phosphates together make something very chemically active And because this thing is so chemically active, when we bring it in here with the correct enzymes, it very, very easily will attach to the new strand of DNA. Okay, so that's a nucleoside triphosphate. It has to lose those two phosphates, those two extra phosphates, and that's called a pyrophosphate that's lost here. And then this T nucleotide will have been added to the chain, okay, matched up with an A. The other thing to note here, if you remember in our description of a DNA strand, there's a 5' end and a 3' end. This would be the 3' or 3', 5' end. And remember, that's based off of, if I look in really closely at the sugar, there's a carbon at each of these points, and then one sticking up here. We label this the first carbon right here, second carbon, third carbon, and so this is the 3' end attached to the third carbon, fourth carbon, and the fifth carbon sticks out of the top, so that's why that's the 5' end. Okay, so we have 3' and 5', and the big thing that I want to note here is that they are showing correctly that the new nucleotide is being added to the 3' end of this molecule, which seems like such an itty-bitty detail, but it becomes hugely important. You're going to see that in the next slide. But this would be at a replication fork, where we would be adding to the 3' end, a new nucleotide to the 3' end, and it complements the template strand or the original strand. I think that's it for that, and we're going to take another detailed look, go a little bit closer at what's happening here. Okay, just an overview again. We're going to be zooming in at a replication fork, so we're actually going to zoom in right here, so it's unzipping, and right behind where it's unzipping, there's going to be new DNA being added. So that's where we're going to zoom in here as soon as I can switch the picture. There we are. Okay, so we zoomed into that replication fork, and just to to point out where we're at and what's doing what, first of all, the dark blue again is the parent template strand. The light blue is the new DNA that's being added. So the origin of replication, if we were to follow this back, might have been all the way back here, and we've got this bubble on top and bottom, and we're zooming in on the replication fork on this side. So there would be a protein, it's called helicase, we'll learn that later, but a protein that would be going along unzipping this, and it's unzipping in this direction. Some things to point out is, again, the 5' to 3' idea. This top strand, if it is 5' on this end and 3' on this end, then its complementary strand is going to be opposite of that. We're anti-parallel. Okay, so if the 3' end on the template is here, then it'll be the 5' here because they run in opposite directions. Okay, so we have this 5' end here, and if you remember, DNA is always going to get added to a 3' end. So if I look at this top strand, if we just look at this, first of all, we've got this pink blob here. That's called DNA polymerase 3, and a DNA polymerase, that name polymer just means to make a long repeating sequence, or something that's a large molecule made out of a lot of little parts. That's a polymer. So DNA polymerase makes a polymer of DNA, and we will find that there's several DNA polymerases. This happens to be called DNA polymerase 3. Okay, well, DNA polymerase 3 is the molecule that's going to add DNA onto the new strand. That's its job, that's what it does, and it has a severe limitation. Its limitation is that it can only ever add to the 3' end of an existing strand. So we've got our 5' end here, this would be our 3' end, and we're going to add on to this. On this top strand, that works beautifully because as this unzips, we're exposing more of the top parent strand, okay? And we can just continue to add onto the 3' end of this. Okay, we call this top where this is happening. It could be the bottom depending on where you're looking, but in this case it's the top. We call this the leading strand, and basically DNA polymerase can continuously follow this as it unzips and make one long strand following it. Okay, this works a little differently on the bottom strand. If I identify my 3' and 5' again, on the parent strand I've got a 5' on this end, 3' on this end, which means in my new strand that's attaching, I've got 3' on this end, and I've got 5' right here, and I'm going to write that in. 5', how do I do this? 5' is right here, okay? And that's a big problem because DNA polymerase 3, remember, it can only add to a 3' end, and here's my 3' end. But I'm unzipping this way, which means as this unzips, I'm not able to add on to this end of things. Okay, so what the DNA polymerase 3 has to do, it's a little inconvenient, but it would have made this section of DNA, detached, come up here, add a new section of DNA, detach as things unzip, come here, add more DNA as this continues to unzip, and you'll see it continuing to zip here. This would have to detach, come back here, and make another segment. So it's going to have to make it in a bunch of little segments. Again, the reason for this is because it can only add to the 3' end, so it has to work that direction. But we're unzipping in the opposite direction, so we have to detach, lay a section, detach, lay a section, detach, lay a section. Anyway, that strand where that happens is called the lagging strand. And what it ends up doing is it ends up making a series of little fragments, because you can see it right here. Here's the first section that was laid, but then it had to detach, and here's the second section that was laid by DNA polymerase. Here it's making its third section, and it would keep going. Well, that makes a little segment of DNA, a little segment of DNA, and so on. Those little segments are called Okazaki Fragments. It's a scientist that discovered them was Okazaki. But those Okazaki Fragments, he discovered those, and that's how they figured out, the leading and lagging idea that it happened that these segments are made. They've also discovered that there is a protein called DNA ligase, or an enzyme called DNA ligase, and what DNA ligase does is comes along and connects the fragments, so that ultimately this is going to be one long strand of DNA without any little pieces, because DNA ligase is going to connect the fragments. Okay, so we're going to kind of pause the description here. This, although it seems detailed, is really a high-level overview. We haven't shown the majority of the proteins that are involved here. We showed DNA polymerase 3, and we showed ligase, but we're going to find out in the next video, 16.2c, that there is a whole bunch of other enzymes and proteins that are involved in this whole process, and we kind of have to sort all of those out. Before I sign off, I am going to say, and I think I said this in the last video, I put 2 animations up on the animation section of the website, and they could be hugely valuable for you to watch as you're going through. Even right now, as you're a little unclear of the 3' and 5' thing, watch those 2 animations. It's going to tell you more information than you need to know at the moment, but I think it'll give you a good visual of what's happening in this entire thing. So that's my advice. The animation section of the webpage is just above where this video is. And there's 2 chapter 16 videos that are in there on replication. Watch them, please. So if you have any questions, you can ask on the discussion board or in class, and we'll see you in class.